everyone, welcome back to another video of STEMAC, and today we're going to talk about bacteria. Now bacteria is the plural form and the singular form of the word would be bacterium. So whenever we are talking about multiple bacteria, we will use bacteria, we will not use bacterias. Okay. So a lot of us think that bacteria are only connected with dirtiness or contamination, because we think that all bacteria are bad, which is a misconception, because there are both good bacteria and bad bacteria, and we call this bad bacteria pathogens. So pathogens. Pathogens means they are disease causing bacteria. So there are some bacteria that help us, like the bacteria that reside on our skin. They help to combat other harmful pathogens. Bacteria can also act as decomposers. They help in our digestion. They also play an important role in nitrogen cycle to fix nitrogen in the plant roots that the plant needs. So they are helpful in many ways. But there are some bacteria that can cause disease, like tuberculosis caused by Mycobacterium tuberculosis bacteria, cholera, salmonella, and so on. So what are bacteria actually? Bacteria are unicellular prokaryotes. So unicellular means they are usually single-celled. And we know that there are two types of cells, right? Eukaryotic cells and prokaryotic cells. Prokaryotic cells are generally much smaller than eukaryotic cells. Our cells are eukaryotic cells. So bacteria fall under prokaryotes. Bacteria are also microscopic. So they are about 0.2 micrometer to 5 micrometer in size. So like they are very small and we cannot see them with our naked eyes. They have a very diverse habitat, which means that they are everywhere, from soil to our skin to even lava. So lava is a very extreme condition, right? So the bacteria that can survive in these extreme conditions are called extremophiles. So they are called extremophiles. Extremophiles can survive in very extreme conditions. Next, they can reproduce rapidly. So there are some bacteria like E. coli that can divide every 20 minutes. Vegetative cells and endospores. Vegetative cells means that the bacteria is in a normal state, so they are actively growing. But there are some bacteria that can form endospores. So endospore is a state of bacteria in which they remain in a dormant state. So they are not actively growing, and they are highly resistant to harsh environmental conditions like heat, lack of nutrient, or freezing temperatures, etc. So how does a bacteria looks like, or bacterium looks like? So here is a basic structure of bacterium, and we can see that we are already familiar with some of this, like cytoplasm, ribosomes, cell wall. Most of the bacteria contain a cell wall, and their cell wall is composed of peptidoglycan. So the cell wall of bacteria is composed of peptidoglycan. Then they have a structure called flagellum. Now this may be unfamiliar to some of you. So this flagellum structure is made up of flagellin protein. And the flagellum structure has a part or portion called basal body, and this basal body attaches the flagellum to the cell membrane of the bacterial cell. And this flagellum works by rotating. They rotate at its base like a propeller to push the bacterium to move in a liquid environment. So flagellum is used for movement of a bacterium. Then we can see that they have cell membrane, and they have another new or unfamiliar structure called pili. So pili is made up of pilin protein. There can be one or more pili on the surface of a bacteria, and pili is required for the attachment of bacteria on other bacteria cells or surfaces. And pili can also help in transferring of genes or plasmids from one bacterium to another. We'll learn more about that later. And then we have circular DNA of bacteria. Now, we say that bacteria are prokaryotes, so that means that they lack a nucleus and they lack other membrane-bound organelles, right? So here we can see that the circular DNA of bacteria, although it is double-stranded, it is in circular shape and it does not have a nucleus around it. So the bacteria is lacking a nucleus, and the circular DNA, it's in circular shape. So now we can also see some structures, circular structures, small circular structures called plasmid. So these plasmids are like extra DNA to bacteria, and they are much smaller to the circular DNA, and they contain fewer genes. So they can contain genes like genes for antibiotic resistance or genes encoding toxins or other virulence factors. And a bacteria can transfer this plasmid to another bacterium cell. So with this, they transfer this gene from one bacterium to another bacterium. We'll also learn about that later. And then we can see that there's an extra layer outside of the cell wall, this blue layer. It's called capsule. And some bacteria can form capsule, or we can also call it a slime layer. And this capsule or slime layer provides additional protection to the bacteria. So this is basically what a bacteria looks like. And then we're going to go to the classification of bacteria. So bacteria can be classified based on shapes, and there are three basic shapes. First is cocci. So these cocci bacteria are spherical shapes, and they can be subclassified into monococci, diplococci, etc. The next is the bacilli shape, which are rod-shaped bacteria, and these bacteria can also be classified further into monobacilli, diplobacilli, etc. And the last is spirilla, which are spiral-shaped bacteria. So just like cocci and bacilli, spirilla can also be subclassified. So another classification of bacteria is gram staining. So bacteria can be classified based on how they react to gram staining test, and it helps to determine their cell wall composition. So there are two types, gram positive and gram negative. Now earlier we learned that most of the bacteria have a cell wall, and it is composed of peptidoglycan, right? Here's peptidoglycan. So in the case of gram positive bacteria, the peptidoglycan layer is thick compared to the peptidoglycan layer of gram negative bacteria. Here we can see that the peptidoglycan is comparatively thicker than the peptidoglycan layer of gram negative bacteria. So But however, the gram-negative bacteria also have an outer membrane outside of the cell wall. So in the case of gram-positive bacteria, when we do a gram staining test, we use a dye called crystal violet. So we use this crystal violet dye, and because of the thick peptidoglycan layer of gram-positive bacteria, they can hold this crystal violet dye and they will express a violet color or purple color. But since gram-negative bacteria has a thin peptidoglycan layer, they cannot hold this dye, crystal violet dye, and so they cannot express a purple color. Now this test is very significant because based on the result, we can determine that which type of antibiotic we will use on which type of bacteria. For like, some antibiotic will only work against gram-positive bacteria and some will work against gram-negative bacteria. So this is the classification based on gram staining. So like all the other living organisms, bacteria can also go through reproduction. So what are the reproduction methods of bacteria? Well, there are two types of reproduction for bacteria. One is sexual and another one is asexual. So first we're going to talk about asexual reproduction, and that includes binary fission. So binary fission is the process in which a bacterium is divided into two identical daughter cells. So here we can see that we have a bacterial cell here with its cell wall, cell membrane, the plastid and the circular DNA. And this bacteria, in binary fission, in this bacteria, the genetic material, which includes the circular DNA and the plasmid, will replicate. So in this second step, we can see that the replication has taken place. So there are two circular DNAs and the plasmids have also replicated. So after this, the cell will elongate. So the cell will elongate, and here we can see that the two DNAs have moved to two sides of the cell along with the plasmids. This is because so that the two cells that we will get after the split, they each get a copy of the DNA and also the plasmids. However, the plasmids will be assigned randomly. So one cell may get more plasmids than the other one. So the third step is called elongation. Because the bacterial cell is elongated and the genetic materials have moved to two sides of the bacterial cells. After this, the cell wall, this is our cell wall, along with the cell membrane will start to grow in the middle. So they are growing, the cell wall and the cell membrane is growing in the middle of the cell. And when these two ends meet, they will divide or split and form two identical bacterial cells. So here we can see that it's a very simple replication method in which a bacterial cell has divided into two daughter cells, identical daughter cells at the end of the replication method. So this is our binary fission, and this is involved in the asexual reproduction method of bacteria. Now we're going to talk about the sexual reproduction of bacteria. Well, bacteria does not really sexually reproduce like human, but they do transfer genes from one bacterium to another. And they do it in 3 mechanisms, and they are conjugation, transformation, and transduction. Now these are horizontal gene transfer. So they are called horizontal gene transfer. Horizontal gene transfer means that the transfer of gene is happening between bacteria of the same generation, whereas the vertical gene transfer means that the transfer of gene is happening from parents to offspring. So first we have conjugation, which is the most common method, and it is encoded by plasmid, which replicates independently of chromosomes. So first here we have a donor cell, and this donor cell has a F plasmid, a special plasmid which is called F plasmid, and this F plasmid has genes that form a sex pilus on this donor cell. So this is a donor cell, and here we have another cell which is a recipient cell. And this cell, as we can see, does not have a F plasmid. So we can call the donor cell F plus cell and the recipient cell F minus cell. Now after this, the donor cell will extend its sex pilus to attach to the recipient cell and form a conjugation bridge. So this is a conjugation bridge. So therefore, for conjugation, it requires a direct contact between the donor cell and the recipient cell. So after forming the conjugation bridge, a nuclease enzyme will break one strand of the plasmid, and that strand will enter the recipient bacterium. So here we can see that the double-stranded plasmid, one strand of the double-stranded plasmid has been broken, and it is entering the recipient bacterium. So after this, the recipient bacterium and the donor bacterium, they both will form a complementary copy of the plasmid, and so the plasmid is double-stranded again. Here you can see that the plasmid both are double-stranded. So after receiving the F plasmid, here we have a recipient cell, and now it has a F plasmid just like the donor cell. So after receiving the F plasmid, we can now call it an F plus cell. And this F plus cell or this bacterium will then donate this plasmid to another new bacterium. And we know that plasmid contains genes, different types of genes, like genes encoding antibiotic resistance or genes encoding toxins or other virulence factor. So when this bacteria will donate the plasmid to another bacterium, it will also donate those genes to that bacteria. So this is our conjugation method, and next is transformation method. So in this method, a bacterium absorbs free genome from the environment. So we know that when a bacterium dies or lies, it releases DNA fragments, right? So this is a dead cell, and it has released DNA fragments into its surrounding. So now not all bacteria will capture these DNA fragments. Only the competent bacteria will capture these DNA fragments. So competent bacteria are the one that will capture these DNA fragments. This is because these competent bacteria have special DNA binding proteins on their surfaces. So these proteins will grab the DNA fragments, and they will transfer the DNA fragments inside of the cell. So once the DNA fragments has entered the cell, they will get integrated into the bacterial chromosome. So here we can see that after entering the bacterial cell, they have been integrated into the bacterial chromosome. And so now the bacteria will express the new genes that have entered their or integrated inside of their bacterial chromosome. And when or if this bacterial cell undergoes binary fission, it will then donate, or it will transfer the gene from parent cell to an offspring. So this is another method in which the gene is being transferred from one bacterium to another. And lastly, we have transduction method. So transduction methods require bacteriophage. So we in our virus video previously, we learned that bacteriophages are viruses that uses a bacteria as a host cell to replicate. So in this case, a bacteriophage, this is our bacteriophage, a bacteriophage will infect a bacterial cell and they will insert their viral DNA inside of the cell. And with this, the bacteriophage will take control over the bacterial machinery and they will force the bacterial cell to produce new viral parts. So here we can see that we now have many different viral parts here along with the bacterial genetic materials. Now what happens is that some of the newly made viruses by mistake pick up the bacterial genetic materials, which includes the DNA fragments and plasmids instead of the viral DNA. So when those viruses gets assembled, here we can see that this virus has picked up the genetic material of the bacterial DNA or plasmid. So when this virus will get out of the bacterial cell after its lysis, they will infect another new bacterial host, and with that they will insert that bacterium genome into the genome of the new bacterial cell. So with this they are transferring the gene from one bacterium to another, and this happens through a bacteriophage. So this is the transduction method of the sexual reproduction of bacteria. This concludes our bacteria lesson, whole lesson on bacteria, which we learned that what a bacteria looks like, their classification and their reproduction methods. And with this, we're going to end today's video here, and thanks for watching.